So, chapter 2 is titled Tools of Environmental Science. And it's not talking about the physical tools, but it's more talking about the cognitive tools. And we're going to be discussing about how environmental science is done and also about how some of the environmental decision making is done. So your book opens with this picture of some scientists looking like they're taking some measurements. It says the scientists are measuring the tail of a tranquilized wolf. What question would these observations help the scientists answer? So what are some of the things the scientists could be looking at by looking at measuring the tail? Maybe the tail tells them something about the age of the animal, or maybe how healthy it is, maybe even like the social status of the animal. So there's a lot of questions that scientists might be asking, and it might start out with some kind of observation like this, or it could be that they already came up with a hypothesis about relationship between something they're looking at and the size of the tail, and they're actually making these measurements to prove or disprove their hypothesis. Okay, so let me just go through the kind of the steps of the experimental method. Again, you've probably done this before in other science classes, especially in biology. I know I go through this for my biology classes, and maybe even middle school. You probably had some of these things. Well, what's the first step in the experimental method, or maybe otherwise known as the scientific method? Is observation, right? You notice something or there's a question being posed. Oftentimes in environmental science, you start out with some kind of a problem and you have to, come up with some solutions. So you have a question or you have an observation. So in this example, you have some students, these very keen students from Keene High School, notice some disappearance of some mussels in their local river, and they're suspecting that something's causing these mussels to disappear over time, and they're suspecting pollution. So that's their question, right? What's causing these mussels to disappear over the time period they're looking at? So they had to come up with a hypothesis. And hypothesis is a testable explanation of their phenomena or their observations. This is what they think is going on, and based on that, they might make some predictions that they can specifically test. So in this case, because they're suspecting some of the fertilizer from the surrounding area is causing these mussels to die. They're going to predict that if they were to expose these mussels to the phosphates, it's going to kill them, right? This explains, what's going on in their observations. So the next step will be to design some experiment, right? And it's really important to understand what a good experiment design looks like, and the difference between a controlled experiment as opposed to looking at some natural experiment or correlation experiment. For a controlled experiment, it's called control because you have something called a control. In experimenting, you really want to be looking at one thing that you're testing. So in this case, these students are testing the effects of phosphate, right? That's the only thing that's going to be varied in their experiments. They're going to try to keep everything else the same except for the thing that they're testing, which in this case is the phosphate fertilizer, right? And the control is something that is the group that is getting exactly the same treatment as experimental, except for the the variable they're testing. In this case, it's the phosphate, right? Oops, what happened? What is not the control group is the experimental group, and this is a group that receives the experimental treatment. So I guess in this case, it's the phosphate. So that's what you're testing, right? So in a good experiment, you should always have a experimental group and a control group, and everything is the same between these two groups except for what you're testing, which is the experimental variable, right? And by the way, the reason why it's saying group, right? Control group and experimental group is because you never test it on just one thing, right? Or one individual or one. There's usually several experimental setups. So let's say maybe in this case, These students probably have several tanks that's going to serve as a control, and maybe several tanks that's going to serve as an experimental, and all the one that collectively will be called a control group and experimental group, because you don't want to just do it on one tank, because if something were to happen to one tank, that's gonna, your results is going to not reflect the actual thing that you're testing, but whatever it is that caused the difference. Okay, so like I said, everything about the control group and experimental group is the same except for the experimental variable. Okay, so when you do an experiment, there are things that stay the same and there are things that are going to change. And the things that are going to change are called variables. The things that are going to stay the same are called constants. So let's say you're doing an experiment, you're setting up some tanks, right? These are tanks. And these tanks are going to be all the same size, same amount of water and all that, except for what you're manipulating. So in this case, you're adding fertilizer in one and you're adding no fertilizer in the other group. Because you're manipulating the amount of fertilizer, this is called the independent variable. You're going to have mussels in here, right? The number of mussels is going to be the same. Let's put 4 at each one. The type of mussel is going to be the same. More or less the size of the mussels, or at least the distribution of the size of the mussels, is going to be the same. All these things are constant. They don't change. What might change is the number of mussels. Some of them might die, right? Because of your experiment. And maybe they don't, maybe only one dies in the fertilizer. So the number of mussels is also a variable in this experiment, but this is dependent on whether or not you added fertilizer, hopefully. So this is going to be your dependent variable. Okay, so your independent variables are the variables that you're manipulating, and the dependent variables are the results, and everything else are called constants. Size of the tank, the number of the number of your groups, the type of mussels, the temperature at which you kept the water, if you're feeding the mussels, the type of food that you're feeding, all that kind of stuff are constant. Okay, and in your experiment, you're speculating that your dependent variable depends on your independent variable, or your independent variable is having an effect and changing the value of your dependent variable.